So next we have Rikeish Muralitharan from Monash University in Melbourne. Rikeish, I can see you're there and your screen has come up. Thank you. And Rikeish will be talking about investigating the role of short-chain fatty acid sensing receptors in hypertension. Thanks, Rikeish. Oh, maybe you need to unmute yourself, sorry. Still muted. There we go. We can hear you now. Thank you.
Thank you for the kind introduction. We all know that a high fiber diet is protective to cardiovascular health. Dietary fiber is fermented into short-chain fatty acids, acetate, propionate, and butyrate by commensal bacteria in the large intestine. These short-chain fatty acids can enter the systemic circulation through the portal vein. Our lab and others have shown in preclinical animal model that a high fiber diet or supplementation of short-chain fatty acids can reduce blood pressure and its accompanying complications by modulating the gut microbiota. Short-chain fatty acids can bind to G protein-coupled receptors GPR41, 43, and 109A. And as we can see here, one metabolite can bind to more than one receptor, meaning that these receptors have redundant roles. GPCRs are expressed lowly in cardiovascular-related organs, but highly expressed by immune cells. Earlier this year, our lab showed that single GPR41, 43, 109A, and double GPR43, 109A have cardiovascular dysfunction compared to wild type, as indicated here by cardiac to body weight ratio, as well as the perivascular fibrosis. But because we know that GPCRs have redundant roles, it is important to study these GPCRs in a triple knockout manner to truly understand their true physiological relevance, as well as truly understand the effects of short-chain fatty acids. Our hypothesis is that the triple GPR41, 43, 109A knockout have a most severe cardiovascular phenotype when challenged pharmacologically with angiotensin II. As for the methods, I will be using C57BL6 as my wild-type control and the knockout strains male and female. I will perform baseline acclimatization and then baseline blood pressure at 6 weeks using tail cuff. Following that, I will implant a mini pump containing angiotensin II for a 4-week protocol and perform weekly blood pressure recordings for 4 weeks and perform some experiments such as the metabolic cages to look at, to investigate metabolism, looking at cardiac ultrasounds, echo MRI for body composition, as well as sodium challenge to look at salt handling. Following euthanasia, I will be collecting organs and tissues for flow cytometry of immune cells, look at 16S sequencing for gut content, qPCRs, as well as histology for fibrosis. So we expect that the triple knockouts will have a more severe cardiovascular phenotype compared to wild-type mice, as in higher blood pressure, more fibrosis in the kidney and heart, more severe cardiac dysfunction, as well as abdominal salt handling. This is based on our preliminary results showing that the triple knockouts have significantly smaller kidneys, as well as that they excreted lesser sodium when given high sodium. Thank you. With that, I'm happy to answer some questions.
Thanks so much, Rikeish. We'll just see. No, there are other questions that have come through. We'll wait for some questions to come through. Please send them through if you have them. I guess I'll ask the first one about the salt handling. Can you explain the, I guess, pharmacology and physiology into how the receptor would affect that a little bit more detail?
Okay, so firstly, so there is no evidence to actually support that the GPCRs actually play a role in salt handling at all. But this is just based on results that we found from the from the study itself. But I believe that it is driven by immune cells, and because they are highly expressed by immune cells, and I'm going to quantify the immune cells and see whether that is related, because there are a lot of studies showing that macrophages play a role in salt handling or drive salt handling in mice.
Yeah. Alrighty. we're still getting questions, I guess, hopefully, that are going to come through. So I'll ask another one about macrophages in the gut. Do you know about the abundance of them in the gut and where exactly they're located, and I guess what layers and what section of the gut as well?
Okay, so in the gut itself, they're actually highly expressed by the epithelial cells. So they are all especially enteroendocrine cells, they express highly of all three receptors, but they are in the epithelial cells, yes.
Okay, awesome. And so Helena has asked, what is a sequence homologue between GPCR 41, 43, and 109A? Oh.
I don't quite understand the question. Like, sorry, Helena.
Okay. Alrighty. I can see that some people have raised their hands now, so I will take to Anastasia. Is Anastasia going to unmute?
Yeah, that was previously. Sorry, that was for the previous presentation, not this one.
Oh, okay. And Brad?
Yeah,
sorry, they weren't coming up on my screen.
I was busy typing away. I was just going to ask Rikesh early on, I think you may have answered about, are they more highly expressed on specific types of immune cells?
So far from the GTEx results that I've seen, they are expressed by all Tregs and as well as macrophages, and especially macrophages have high expression of these receptors.
And I got one more question here, Grant. Why did you choose the Ang2 model as opposed to a salt-sensitive model of hypertension?
So thank you, Grant, for the question. So the reason was actually the Ang2 model is something that we have been working with, and we did not know how the mice would have actually responded to the salt challenge in the first place. So in future, we might look at different models, whether it can affect the salt. Yes, thank you.
Yeah. Thanks so much, Rikesh. I'm so sorry for that technical difficulty, everyone.
Thank you.
Yes.